is Jordan and this is Evelyn, and we would like to test the effect of the addition of nutrients on the growth of soybean plants. So just some background information, there are two different types of nutrients needed for plant growth, and those are macronutrients and micronutrients. Macronutrients are needed in larger quantities, and micronutrients are needed in lesser quantities. So the macronutrients are hydrogen, carbon, oxygen, nitrogen, phosphorus, potassium, sulfur, magnesium, and calcium. There are three bolded. They're kind of hard to see right now, but it's carbon, nitrogen, and phosphorus, and those are the ones that we would like to test. The micronutrients are boron, zinc, manganese, iron, copper, molybdenum, and chlorine. So there are also two different types of macronutrients, and those are primary nutrients and secondary nutrients. The primary nutrients are nitrogen, phosphorus, and potassium. These ones are usually lacking in the soil, so if soil needs fertilization, it's normally of these nutrients. The secondary nutrients are calcium, magnesium, and sulfur, and these usually have enough in the soil, so these are not needed to be fertilized as often. Calcium and magnesium are added when lime is added to acidic soils, and sulfur is found because of the slow decomposition of organic matter in the soil. So nitrogen is used to help plants with rapid growth. It increases the seed and fruit production of plants, and it improves the quality of leaf and forage crops. It's also a necessary part for the transfer of energy. Phosphorus is involved in the formation of all oils, sugars, and starches. It's essential for photosynthesis. It helps transform solar energy into chemical energy, and it encourages blooming and root growth. Carbon dioxide increases the production of fruit, it increases the rate of growth for most plants, and it increases leaf photosynthetic rates.
There are other studies that are similar to the one that we want to test. We ended up picking 5 different tests. The first one tested elevated carbon dioxide on mangrove plants. They separated the plants into 2 different groups. One was placed into a glass house with 400 ppm of carbon dioxide, which is the natural level of carbon dioxide in the atmosphere. The other group was placed into a glass house that was at 800 ppm, and they maintained this by using high pressure cylinders, which kept it anywhere from 790 to 810. They ended up showing that growth increased by 42% and photosynthesis by 115. The root volume also doubled. Plants with elevated CO2 grew more than the normal level. However, plants that had both added nutrients and elevated carbon dioxide grew the most. The other study that we picked tested nitrogen on different stages of growth of oilseed plants. They ended up measuring biomass and seed yield. There was 4 different stages that they applied nitrogen to. The first was the very beginning, and the second is when they grew 5 leaves, and the third is when they grew 9 leaves, and the fourth was when the stem started to elongate. This showed that high application that was not above 135 kilograms were the most successful. It also showed that nitrogen increased biomass and seed yield. The other study that was tested, was the response of different irrigation and nitrogen levels on red pepper plants. The plants were divided into 2 groups for the irrigation part, and another 4 groups were divided for the nitrogen levels. The nitrogen levels were at 0, 80, 160, and 240 kilograms. It also proved that biomass and the fruit weight increased with the increasing levels of nitrogen. The other study that we used tested phosphorus on a meadow. They added the phosphorus solutes by using a watering can. Then they measured the dry weight of the plants. This research also stated that they needed to be done more because it proved that phosphorus did not have an impact on the plant, but I think that this could be that the soil already had a high concentration of phosphorus and that the addition of more phosphorus had no effect on the plants, since they could only absorb so much of it. The next study that we picked studied the effect of water and nutrients on resprouting trees. The trees were exposed to different levels of water and nutrients. The nutrients consisted of both phosphorus and nitrogen. The study was conducted for 2 years, but within the first 12 months, the shoot diameter increased, along with the shoot length and the re-sprouting ratio. It also showed that trees with higher concentration of nutrients responded more than the ones with lesser.
So this chart shows the average amount of soil nutrients and what we would like to test. For nitrogen, the average is about 5 to 10 parts per million. We would like to go below, use the average, and then also above the average to show both sides of the spectrum. So we want to test 2, 5, and 10. For carbon dioxide, the average in the atmosphere is about 400 parts per million, and we would like to test 200, 400, and 500. For phosphorus, the average is about 10 to 20 parts per million, and we would like to test 20, 30, 40, and 60 parts per million. And here are sources from where we got the average numbers. The question that we are trying to answer is, do nutrient levels influence the plant growth? And our hypothesis is that the addition of nutrients in the soil and atmosphere will affect the growth of plants. We chose the soybean plant because it is widely used, and because of the slow increase of carbon dioxide in the atmosphere, we wanted to see how that will soon affect the growth of plants. For our method, it's broken down into 3 sections. For carbon dioxide, we want to use 150 seeds broken up into 3 different pots, so it's 50 seeds in each pot, with the concentrations of 200, 400, 500 parts per million, and the pots will be placed in a sealed chamber that is controlled with the concentrations of the carbon dioxide. For nitrogen, we want to use 100 seeds in 4 different pots, so that's 25 seeds in each pot, with concentrations of 2, 5, 10, and 15. And the pots will be placed in an incubator at 22 degrees Celsius. For phosphorus, it's very similar to the nitrogen, which we'll be using 100 seeds into 4 pots, which is 25 seeds each, with concentrations of 20, 30, 40, and 60 parts per million, and the pots will also be placed in an incubator at 22 degrees Celsius. After the growth period of 21 days post-germination, the plants will be dried to 70 degrees Celsius, and we will measure their dry weight.
There are 3 different components of our experiment. The first is the control, which are the ones that are kept constant. And the second are the independent variables, which are the ones that we manipulate. And then the dependent variables, which are the ones that rely on the independent. The plants will be kept in an incubator at 22 degrees, and they will be exposed to light 12 hours and then dark for 12 hours again. The moisture will also be kept the same for all the plants. And they will be originally exposed to the same amount of nutrients in the soil, and the ratio will be at 5:5:5. And we will measure the growth after 21 days past germination. We did that because the seeds already have nutrients for the embryo, so the surrounding nutrients in the soil wouldn't affect the growth. Our independent variables were the concentrations of the different nutrients, and our dependent variables was the plant growth and also the dry weight of the plants. For nitrogen, we expect above 5 to improve plant growth. We think this because 5 is the natural level of nitrogen in the soil, and other studies also showed an increase of the growth. For carbon, we expect plant growth to increase with the elevated levels that are past 400 ppm. Other studies showed that high applications of carbon dioxide also increase plant growth. And for phosphorus, we expect for plant growth to slightly increase with the levels of phosphorus. Phosphorus is a major nutrient that plants need, so it should help increase plant growth, even if some studies do not show this trend. Here are our references, and are there any questions?
Thank you.